something new.
Yeah. I think it will be fine. Window full screen. F11. No. Where is it? Full screen? Yes. Oh, that is on full screen. Okay, fine.
It doesn't matter. Admit all. Someone maybe from MRIs.
Oh.
What is this?
Some security.
Yes. Vault. Will it be closed?
No, it will be open. The meeting?
Uh-huh.
Okay.
Here only this meet. Now they have to admit it.
Oh, yeah. How do you set the setting every time admit all?
Okay, I will join from my. Do I have the access to admit?
Yeah. I
Or
gave
do all
you
people
the
have?
editor access. Let me start sharing the screen. There are some settings I'll upload. Recording. I should admit you.
Only 1 minute. Cancel.
Is my screen visible? Okay, I think I have to escape this and then do this.
Yeah, I can't open with my eyes ID.
Should we? On the
Okay, we should start, I guess.
That is your camera. Say recording.
I'll join and you admit me and I will then keep admitting other people. I will keep admitting them. Okay. It's
I'll
off.
make you.
Yeah, it's off.
The timer will start.
Yeah.
Wait.
Can you make me like some here? Make a co-host. Okay. Yeah, that mark works.
Should we? Can you see the screen?
Yeah, yeah, we can see this.
Okay.
Mm.
Okay, I should start my mic. Okay. We'll start. How many people are there?
15.
Okay, we'll start. So, hi everyone. Uh, is everyone there here? Whoever joined?
Yes, sir.
Okay.
It will be really great if we can see you, if you are in a place where you can turn on your videos, and you can keep your microphones on if there is no noise around, otherwise it's fine. So we also get feedback like if you are uh, audible or not, and you are able to see your screens or not. So yeah.
We'll start. Okay, welcome everyone. Today is the first day for the introductory class for neuroinformatics and methods in neuroscience course. So, without further ado, let's get started. Should we wait for a few minutes? No.
I think it's time.
Okay, we'll start. So, welcome to the course. We are the instructors for the course, me, Deepak Raya, and Atharva. So, we are PhD scholars in Center for Neuroscience at IISc Bangalore. We want to name ourselves as not instructors, but co-learners, because we are learning with you together. So we will just be a guide to play with the neuroscience data, etc., and some revision of concepts. So just a brief about ourselves. And firstly, I'm Deepak Raya. Uh, I'm working with Dr. Sridharan Devarajan at Center for Neuroscience in IISc Bangalore, where I study the neural basis of interaction between attention, working memory, and eye movements through various neuroimaging techniques. So my background is in electronics and communication engineering uh, from Chaitanya Bharati Institute of Technology from Hyderabad, and then I did my MTech in signal processing from IISc Thiruvananthapuram. So this background helped me a lot in analyzing neuroimaging and other imaging data. So next, over to Atharva.
Hello everyone, I am Atharva Modi. I did my BTech in chemical engineering from IIT Bombay in 2019, and then I was working in a cigarette industry as an assistant manager, and then I took a leap of faith to join neuroscience. I was at NCBS for a year, and then I am currently doing my PhD at Indian Institute of Science, Center for Neuroscience, in Dr. Ashish Davle's lab. And from that leap of faith, currently I am studying how do we learn to make the right leaps by studying the neural basis of trial and error motor learning, and I use rats as my model systems. So I am another co-learner apart from Deepak in this course, and um, yeah, we'll go through. Today we'll cover like some introduction about neuroscience, and towards the end of this lecture we'll cover what like where this course fits in an overall broad scheme of neuroscience, and uh, what are we going to cover tentatively. What is our plan, and we urge you to give inputs as well. Like what do you want, because we want it. As per your needs, this course should be as per your needs rather than one side conversation. So please be active in this lecture too.
Okay, sir.
Okay. Uh, so let's get started now with the. So what do you see here is a three-pronged gelatinous wetware, which is known to be a most complex organ in the universe. So Can anyone tell me how many cells are there, neurons are there in the brain? Approximate
In
number.
human brains, I think
Like
it's about 86 billion neurons or something.
yes, 86 million.
Yeah, great. That's the right answer. Average human brain has around 86 billion neurons, and that is why it's also complex to study the interaction between these neurons. So these around 100 billion neurons, and each neuron sends around 10 to 100 electrical pulses to other
Every
neurons
seconds.
per second, and to other 10,000 neurons approximately. So at this complexity, it is known to be a most complex organ in the known universe, and of course, it's the seat of all the intelligence and interpreter of all the senses, um, and also initiator of all the movements and controller of all the behavior in your day-to-day lives. So it's such an awesome thing to study the brain and understand its the underlying principles of it. Okay. So also, brain is very energy efficient. If you compare brain energy consumption, it only consumes 20% of body's energy, and it uh, calculates up to 20 watts a dim light bulb, and whereas at the similar intelligence or the new computer systems which are able to perform this artificial intelligence algorithms and computer vision, for the simple and as well as complex tasks, they require far and far more energy compared to the brain.
For example, a
So,
typical laptop that we have is around 60 watts, and it can do tasks which are um, like much more efficient than even human brain, but there are certain tasks in day-to-day life which human brains excel at, and currently the machines have not reached that level. So this is another aspect of why brain is fascinating. uh, yeah.
yeah. So, until now, anyone has any comments here or any doubts you can mention or any questions? Okay. So now, because of its awesomeness and the things it helps us to achieve, it is of general interest. But before that, saying it is energy efficient, also we have to be little cautious that for a given body mass, it is metabolically very expensive because of its sheer complexity and its involvement in our day-to-day life. It needs this kind of energy consumption for a given body, but when you compare with machines, it is far less for the similar type of tasks that we do. So, for example, brain only has it is only 1.9% of the body's mass, but it consumes almost 21% of the energy. Even like all the other tissues combined just consume only equal to the brain energy consumption. So it is just a caution to say that it is also expensive and energy efficient compared to the machines. Okay. So now these brains enabled humanity to achieve breathtaking milestones, such as exploring space in science and technology, mapping the whole human genome, science and technology, electronics, mathematics, literature, art and crafts,
Basically
etc.
everything.
Yeah, really everything that we
So
achieved until now. And this list goes on and on, and one slide is not sufficient for that. So, because of this complexity and very what do you say interesting phenomena that we can do with the brains, it has interested many people and it has been of general interest since ages. For example, here you can see the Egyptian medical papyri, where it belongs to 17th century BC, and the word brain was first mentioned here. Although the exact word was not mentioned, it is the infographic kind of thing that was mentioned there. And since then comes to like first century AD, they have been describing in general physical description of the brain in various anatomical textbooks in those times. And in the Middle Ages and Renaissance, Leonardo da Vinci has drawn the anatomy of the brain, skull, etc. of dissected, and then it interested many people, and then so many people went on in that Middle Ages and Renaissance to try describing the brain and its phenomenon. Here you see Descartes' illustration of mind-body dualism, and here he describes how the sensory input goes and drives the soul of the body. And since then and now, we also are interested in studying the brain. So at those times it is only studying the physical aspects like anatomy and all, and slowly we want to understand the mechanisms and principles. So there were very early recordings of this nervous system happened by done by Galvani in 1790s, where they recorded the electrical impulses from the frog. And here in 1924, coming like over time, there have been many recordings or seeing the electrical impulses in the nervous system. But now, in 1924, this was the first EEG recording done by Hans Berger, also known as father of EEG. And then most famous and Nobel Prize winning studies from Hodgkin Huxley, where they studied action potentials from giant squid axon, and Hubel and Weisel, phenomenal discovery in how visual neurons respond to the world using the cat's brain and some oriented light bars. So this is the general history of how it happened over time and how people are interested in studying the brain. And in my opinion, everyone is a neuroscientist because in our day-to-day phenomena, we always introspect about our own behaviors, and we try to understand about ourselves, and that way, like, everyone is interested in the brain, and it is very important because it's one of the crucial organ in our day-to-day activities. You have any comments?
Yeah, means we don't have access to the techniques that are available to us in labs. All of you don't have that. So
Yeah.
our goal is today is to introduce you to general techniques and where they fit in. What can we study using those techniques, and where will this course, what will this course cover amongst those techniques? So yes, next probably. Yes.
Yeah, so since then, neuroscientists or scientists across various disciplines have been interested in various questions. And just to give a broad overview of different questions, so the first basic question that people used to ask is, what is the structural and functional organization of the brain, and how does it work? How this mere electrochemical signals traveling across the brain tissue lead to our day-to-day complex behaviors and cognition? Uh, how is this seamless sensory motor control implemented in the brain, and how are memories and information organized? Yeah, sorry. Yeah. And what is the physiology and mechanisms behind various mental neurological disorders? Although we are saying and putting brain in a very good view, but after all, it's another organ or a machine. So brain is also prone to errors and at different scales and different times, and it's also of our interest for the general good to understand these disorders. So overall, like, these are few types of questions that neuroscience and neuroscientists try to answer. So now, recently with the so in the past we have been limited by the way we can read the brains, but now with the advent of technology and advanced computers, it has enabled us to record and study the brain at scales, and that is how the uh, neuroscience has grown so much nowadays because of this technology and computers. So, for example, the uh, revolution in optics and optical phenomenon has introduced this genetics has introduced this fibre photometry and recording the neural responses, and the advent of MRI has helped us to record the brain and its activity and its structure non-invasively. Just you put your head into the MRI and you get your tissue contrast, etc., and even blood activity. Uh, then we have EEG and introduction of different types of electrodes and nanotechnology has enabled us to record brain at scales, the electrical responses, as well as uh, the advent of micro cameras and microscopes has enabled us to study the brain at different scales. And last but not the least to mention is the advanced computers and storage devices which enable us to store this deluge of data and make sense of this. So this has enabled us to study brain at multiple scales, and here you see a depiction of that at what scales you can study. Where, for example, you can study the connectomes of the brain using the MRI. This is a diffusion MRI image where it shows the how the fibers are connected across the brain, and microscopy and uh, at circuit level
And physiology,
yeah,
electrophysiology.
electrophysiology has helped us to study how different regions and different population of neurons interacts, and which gives rise to like different neural circuits interacting with each other. And then we can also image, it's called spatial transcriptomics, where you can in real time see how the protein structures and RNAs express themselves in the brain during different, either cognition diseases, etc. So yeah, and just an interesting picture that I have gone through to see the parallels. So here we have seen like a lot of people are from bioengineering or engineering background, and few are from medical background. But these are just interesting parallels in engineering. So you can, as we have shown previously, we can study brain in different scales.
You can't see your cursor.
Okay.
No.
I'll put one second. Sorry. Yeah. I'll put. Yeah. So, at different scales, so you can study at the whole brain level, then you can study at the population or surface levels, extracellular levels, more deeper at the synaptic levels, between connections between the neurons, and at the cellular levels, what are the membrane mechanisms, how are this voltage is conducted. So a parallel drawn with that, on the right you see the analysis levels where at the whole brain you can use EEG, fNARS, it's a near infrared imaging, fMRI, to study the brain non-invasively at large scale. Then you have cortical electrodes and microelectrode arrays which can record from depth. Then at cellular levels you have these recordings. And similarly, when you draw parallels in engineering, as you can see here on the right side, you can study at the whole systems level, and then you can have a logical map level, and so on. You can go at the very bottom silicon level and study how the components in an electronic circuit interact. So this is one of the interesting thing, and I want to also mention a study or a study actually. So it's called "Could a Neuroscientist Understand a Microprocessor?" It's a very interesting study where using the levels at which neuroscientists analyze the brain, they have used exactly same techniques to analyze the a microprocessor. So it shows a very close parallels between it, and this is just an interesting thing that I wanted to mention how uh, we can use neuroscience for other purposes also.
But their answer was negative, right?
Huh?
Their answer was right that
Yeah.
we can't understand a microprocessor the same way that we are trying to
Yes.
understand the brain.
Hmm.
So probably we need some other
We need some
ways.
yeah. So
even-
yeah, if interested, you can go through this paper, or if time permits, we can discuss. And want to say this?
Yeah, I can. Yeah. So now we introduced many techniques, or Deepak introduced different kinds of techniques, right? From using optical tools or magnetic or electrical tools. But where do they fit in? So if we plot them on a 2D map, where on the y-axis you see the size or scale on the spatial scale that we are able to record in mm, and on x-axis you see the temporal resolution that the technique can provide us to record the brain activity. There are different kinds of fields that you get from each kinds of techniques. For example, electroencephalography, EEG, or magnetoencephalography, that is used for recording brain. So that fits in somewhere here, which gives us very which can allow us to record a large part of brain, but with a relatively poor temporal efficiency.
Good.
Good temporal
Dynamics.
precision. But similarly, fMRI, which can cover even like minute details in the spatially, but it cannot give us that kind of temporal resolution as EEG can give us. So if you want to record details of the brain, uh, like you want to probably work out with the volumes of 1 mm size scales in the brain, you would choose fMRI and not EEG. But fMRI has its own con that it does not provide the temporal resolution. Then these two methods are non-invasive. There are other invasive methods which involve like single unit recording or microscopy, both in vivo and ex vivo, like outside the animal and inside the living animal. You can do both kinds of things. So this patch claim technique, this can provide us a resolution of cellular level, like each cell you get activity from individual cell level, and it can provide a lot of breadth in the temporal resolution as well. So you can record activity of each particular neuron, but how many neurons will you record from? So this does not, like, you can record from individual cells, but not many cells at a time. But there are other kinds of recording which can give you single unit recordings. So basically what it does is you can record from a large number of cells, and then post hoc, like after recording the data, you can analyze that, or even live that, and you can divide them in single units. Basically you can using the data analysis tools, you can identify if the signal is coming from particular cells or different cells, and you can assign units to each activity with. And units here is term, like, is a proxy term for cell. I am using a very rough term for now, but that's what we understand from it.
There are some doubts you can ask.
Okay. There are some doubts on the chat. You can choose to speak, or
Please ask your doubts here. You can introduce yourself and ask a doubt, or directly ask the question also, it's fine. Okay, we'll see the chat.
yeah. So basically, as you go down, as you want to increase the spatial resolution, it becomes more invasive kind of techniques. So what we are going to cover in this course is these 4 techniques, where Deepak will cover EEG and fMRI, data collection and analysis, and I will cover calcium imaging, or it's not exactly imaging, but it is a photometry. It's not imaging per se. Like, I will not go into a cellular level resolution. I will be here somewhere. I will be in the size of 1 to 0.1 mm kind of size scale. I will not go down. So this, not the whole square of calcium imaging, but a part of it. And also we'll cover the analysis part of how do we get single units from extracellular electrical recordings from the brain. So this is what our plan is. It covers a broad spectrum on this plot, and we hope that you get to like something about in the end, and you are comfortable with the analysis. So yeah.
So as Atharva told, we will be covering broad spectrum by covering EEG, MRI, and then at the cellular levels, electrophysiology and optical photometry. So as we have seen now, like, nervous systems generate complex responses. So you see here is the time axis on the x, and y axis is the different neuronal units, and you see how complex is the response. So each dot represents the firing of the neuron, and the color represents the density of firing. So you can always see a density of firing here, but you can't make actual sense of it just by looking at it. Also, when you take other modalities, this is the calcium imaging. where fluorescence of the neuron activity is captured, and here you see their time series, and this is very complex to see. And then you have EEG responses here as time progresses across different electrodes on the scalp. You see how complex it is to the naked eye. You can see some oscillations or some prominent oscillations, but not really make sense out of it just by looking at it or visualizing it. Then here comes the bold response. As we have mentioned, you can get a better scale of space, like which part of the cortex is active at a given point of time, but you lose a temporal resolution because it's not exact neural dynamics, but it is the blood oxygenation level that is going to when a part of brain is active.
So for those those of you don't know, this bold response is a response that is captured by fMRI, functional MRI.
Yeah.
That we are going to cover, and each of these plots is like not easy to capture. So the first one, if you see the activity and units on the y-axis, so I will tell you like how do we obtain that from recordings.
Yeah.
And each of these have like some common principles at the end. Like when you capture data, they have a very common principles, and even in like in operational considerations, like how do you align them to behavior, how do you study both behavior and these together. So these kind of things we'll try to cover. So we'll not per se cover calcium imaging, but the principles more or less remain same across. Once you try to understand, you will get it very quickly if you have learned it previously, some other technique.
Yeah, the
So-
point we are driving here is even to get this visualizations, it's not a straightforward process.
Yes.
So you have to classify which unit it is coming from, and then there are so many filtering, processing, pre-processing techniques. So this is a DMRI image where you can see the flow of water molecules and map the connections between different regions of the brain. And this is microscopy imaging, and also you have at the molecular scale here. This is a spatial transcriptomics. In real time, you can capture how the proteins and protein transcription changes. So this way, the main point is visualization helps to appreciate the complexity, but the main question is how do we make sense of this. So that is what this course helps you all in, like how to make sense of neural data, and mainly we'll be focusing on this electrophysiology, EEG responses, bold responses, and
photometry data.
Photometry data, which almost looks like this calcium imaging, but it's slightly different, but the principle being the same. So also there is a deluge of neuro data like across different modalities, as you have seen different modalities, and it is very complex to the naked eye. So how do we make sense of it? So this is where different subclasses of neuroscience comes in. First, the neuroscience comes in where we use method of scientific inquiry to understand brain behavior correlations and mechanisms behind various phenomena. We form the hypothesis and then test. And then we can't just test by making hypothesis. We need some way to do experiments and record the brain activities. That is where techniques and technologies to record brain comes, and data processing and analysis technique comes. And that is where the part of the course called methods in neuroscience is going to help you guys. And then we have neuroinformatics, where recent advances in statistics, signal processing, and AML has enabled us to quickly make sense of all this complex data and make inferences on it. So this is the whole scenario of this neuroscience, methods in neuroscience and neuroinformatics. And right now our course is as you know already, it is named as neuroinformatics, where we'll be learning different techniques to analyze and process the neuroimaging data, and the same goes with methods in neuroscience. Also, sometimes we need to integrate data across multiple modalities. There are studies where they integrate basically in clinical scenarios, where they integrate blood chemical levels and other phenomena, heartbeats and gut responses with the neuroscience data. So that is another level of even complexity to make different types and different types of data. And this is where neuroscience and neuroinformatics or any of the related umbrella term needs an interdisciplinary and open approach. And nowadays there is a lot of open source data available, so we are going to use this kind of open source data and uh, try to be comfortable with the methods and learn the existing methods. So yeah, this is a nice infographic showing the either neuroinformatics or methods in neuroscience in a nutshell. So here you can see we have a brain or a tissue either in humans or any other animal models, and then we have with technology advancements we have many recording techniques, and then we have used computational power and mathematical techniques to analyze the recordings and make sense of it, and then we've tried to use statistics to find the structure in the data and inform our scientific findings. So this can lead to scientific insights as well as clinical insights, and these are all related together in the long term or bigger picture. And this helps us also in understand how the intelligence works and what are the mechanisms behind it, and these are also useful in optimal experimental design. So you see, you record, you make some processing, you see certain patterns, and you'll also be informed of what hypothesis to make of the data. It is more of an exploratory and exploratory design where even this kind of things helps there. And nowadays you have been hearing about neuroscience inspired AI a lot of times, so that is another domain, and this is a whole big picture in a nutshell. And if you're interested, you can read this paper and get to know even more deeper explanations of each of the stages. So any doubts until now? Okay. So now we will discuss certain course logistics, and towards end we will see some real-time examples of how neuroinformatics or methods in neuroscience can help us in general. So over to Atharva for the course logistics, and we can discuss together. So if you have any doubts or any clarifications, please ask then and there, so that we can clarify or take your opinions. Okay. Yeah.
So the course goals include understanding fundamental principles and practicalities behind most common macro and micro scale brain imaging techniques. So brain imaging means not imaging like using an optical microscope or something, but recording brain. What we meant was recording brain activity on a macro or micro scale in different forms. It can be electrical, it can be activity related to the blood levels or blood oxygenation levels, or it can be activity related to neurotransmitter concentration inside the brain. So we'll cover through these 4 techniques these kinds of the practicalities and fundamental principles behind the techniques. Then we also wanted you to get acquainted with data structure and formats of various techniques that they give output. So the goal is to how we worked on some of these techniques ourselves. So we'll share like how did we make our own setups, and how did we set up our own pipelines, and then we'll also show, as we have done it, that integrating neurodata with behavioral responses. So how do you do that? How do you study both of them together? And as per your requirement, we will try to explain you the mathematics fundamentals behind each of these analysis that we do, or even in the techniques. When we set up, what kind of fundamentals go behind them. So and in the end, we'll also try to get you acquainted with existing tools and techniques in the context of neuroscience, and how are they being used in a creative and novel ways. So this is the course, like the goal of the course looks like. This is what it looks like, and the approach that we are following is the lectures. In the lectures, we'll cover principles and concepts behind various tools and techniques. So there will be dedicated lectures for each techniques, which will introduce you to the principles, how does it work, and how do we set it up, the hardware and the software to record the data. And then we'll also have code and code notebooks to analyze to show you that how this analysis is done when we record. After we record the data, how does the data come in? How do we make sense of it, and how do we make it? How do we clean it? How do we make it analyzable? And then what kinds of analysis techniques do we use? So a lot of things go before the recorded data becomes even analyzable, and then a lot of things go, a lot of math goes behind analyzing them to, in the end, like to make plots where you can visualize, where you can make sense out of it. And some data are like totally you can't even visualize them. So what we are going to cover is that a hands-on approach where we'll share notebooks with you, and you can like we'll show you ourselves first, then you will have some doubts about the fundamentals of them, like how does it work, then we'll cover that, your doubts basically, and math behind it, and then in the in the end of those, towards the end of the course, we'll have another we'll present you the same code, and we will our goal is that you can create your own notebooks, similar notebooks, and you can analyze the data once it's given to you. So that's the goal basically. And there, like, if time permits, we'll share some case studies with you, and maybe some pop quizzes or open quizzes once in a while, not to grade you, of course, but just to keep it interesting. Even you can make some on your own. Then, so another important part of these online course is that these all lectures are recorded. Online availability is in hands of the IEEE platform, and it's uncertain, so we can't guarantee that all these lectures will be available. So we advise you to attend the live sessions as much as possible and make it more engaging, not like what's like we want feedback from you, so our brains also work on inputs from you. And we urge you to maintain like professional conduct throughout the course. That's a given, like without mention it should be taken.
It's like a two-way learning. We are also learning from you. That is why we named ourselves as co-learners, but not instructors. So if you have any inputs over time, or if you're experienced with some type of data, you can also give inputs, and we will happy to take the feedback and get back to you. So yeah. So as again repeating it, that it's a two-way learning, and it has to be like more like a discussion than an instruction.
Not destruction and instead of instruction, but discussions.
Yeah.
Don't destroy anything.
Yeah.
So course timings that we decided on was Tuesday, Friday, 6:30 to 8. So we also have labs in the afternoon till evening. So we are only available in the evening time, and Tuesday, Friday seems logical, so that you also have gap between each lecture, and we also have gap between each lecture to prepare.
Okay.
Yeah, if somebody raised hand, Mr. R.K.
Yeah,
Goyal.
R.K. Goyal.
Yes.
You can unmute yourself and speak.
Yeah, so is it possible that you, instead of Tuesday, you conduct this session on any other day? Because I mean, there is a clash with another course, and I'm very much excited for this course, and I would be attending all the sessions, but neither I do not want to miss that course also.
Okay, so we are happy that you are very excited about this course. We will actually put a poll in the Slack channel and let you know, because in the previous poll we have received that most of the people are comfortable. But we can put we can put the poll again and get to know. But also keeping the conditions that we have in our labs, this is the best time that we got in conclusion. But
Probably like we'll give a choice on Wednesday.
yeah,
People, that also seems fine, and
Yeah.
if nobody has problem, we can shift on Wednesday. But since this was communicated to everybody, we'll again ask everybody that if anybody has problem on Wednesday, and check
Okay.
it after this class.
we can ask right away. I think.
Yeah, if few people have problem, they can watch the recorded lectures, right?
No, that's not certain. Like
Yeah, we don't know when they will be published, so they might have to like in order to come for next class, they might have to again check it. So, Hamid, there is another doubt. Prasoon.
No, hello. I don't have a doubt, but yeah, this timing is not very suitable for me either.
Okay.
It's my office hours, so it'll
Oh,
be rather difficult.
so is Wednesday fine?
No, it's office hours.
I mean, sorry. There are 2 days work from home, so I'll have to adjust which 2 days I can accommodate. So I'll have to look forward to that. Typically, but I haven't yet decided that.
Okay.
But why not a weekend? I was just wondering, like, is weekend not an option?
Uh, Weekend is an option, but I don't know if everyone is comfortable. So we'll put a poll in
We can
the
explain.
yeah, we'll put a poll in the group and let you know. Okay. So yeah, otherwise it becomes too difficult right now. So we'll put a poll right away after the meeting, and then we will let you know if we can shift or change the timings. Is that fine? Yeah.
Yeah, thank you so much.
Okay.
Yeah, thanks a lot.
Yeah. So, as we have communicated to you already, these are the course logistics where we have meeting link and the course Moodle page. And I heard some of them are not yet added to the course homepage. So it might take some time because it's not in our direct control. So uh, we will try the team to let them know about the new people who are not being added, and then we'll get back to you. And we also have for easy communication, we have a Slack channel where we can directly message or post doubts, or we'll post some material directly there in case the course page is not accessible for everyone. And if you have any queries or anything related to the course, please email us, either me or Atharva, or you can email both of us. So this is a course logistics. And coming to the tentative plan, so since this is a mixture of two different levels of courses, like different brain imaging techniques, so we have divided among ourselves on different days or weeks accordingly. So we will post the slides in the group, or we'll send it to you, and you can go through the things. If people are interested in both of the courses, they can attend all the classes, and we are happy for that. But if you have some restrictions and you only have interested in particular domain of neuroscience or neuroimaging data, then you can choose your days and come for that. So we have made it flexible, but and we have seen that many responses are like everyone is interested in both of the courses.
Yeah, more than 85%
Yeah.
people are interested in both, learning both parts aspects of it. And also it's not like Deepak will not cover
Yes.
something that relates to fiber photometry or electrophysiology. So any of us can cover
Yeah.
both,
Yes.
like mathematics behind both kinds of techniques. So for example, on week 3, if you see like signals and systems, time frequency representation, and Fourier transforms, so Deepak is covering that, but this is as useful in fiber photometry as it is in fMRI or EEG data.
Yeah, it is not
So-
fully independent. So,
so-
we have made such a way that we cover the concept before, and accordingly the notebook comes afterwards. So it's not two independent courses. So that way it would be very advantage if you could attend all the classes, so that you don't miss the connection or the theory or mathematics behind it. So you can go through the tentative plan here. So yes.
Anmol.
Hello. I just want to ask, like, what will be the prerequisites of this course? Like I can see, like, in week 8, we are going to do like ML, right?
Yeah.
Dimensionality reduction
So
and all.
we will give the fundamental, and we will give like good analogies, so that people who are not experienced with ML or signal processing can understand it. So this is a part of ML. It's not full ML. So if we take ML course, we have to take another course.
Not particularly about ML, but I'm talking
Yeah.
about the like whole course.
So
Also, it will be
yeah.
beneficial if you give us like the
Yeah.
resource.
So we will send the resources either on Slack, where you can learn the like you can go through the fundamentals, and in the class also we will actually give a fundamental picture of it first and then go through the concepts. So if you have any special doubts or anything, we can always meet or individually meet and also do the basics. But we will send you some very good resources and talk about the fundamentals, so that shouldn't be a problem. And
Means the thing here is that if you like if I want to explain the analysis, I can even explain it in 2 sessions to a person who has already been through these kinds of analysis before. So it depends, and somebody who has not is not familiar with math or these kinds of analysis may take a month to understand this, and we know that there are people from both kinds of background in the course, and that's why we plan to have a tentative plan like this, and we also will also share in advance that what we are going to cover in the next class, and based on that you may choose, like do you want to attend and get involved in the lecture or not. And like if you decide to attend the lecture, we highly recommend that you be engaged, and if you are having any kinds of doubt, however small they seem to you, you can ask them. And if anything, if we feel that okay, this will require more explanation, we'll get back to you for sure. So with after the class or during the class, we can make another room or something like that, or we'll share some kinds of resources on Slack. But don't feel that I don't have an ML background and that's why I can't understand dimensionality reduction. It's a way, like we'll try to give an intuitive. So what happens that in the notebooks, when you will see these will be one line commands. So when you will apply like a filtering of signal, it will be a one line command in the notebooks. So nothing is hampering you from applying those commands, even if you are from a humanities background. But understanding math will like not give you formulas or something, but keep it more intuitive and give some math into it so you can understand it. So that's how we'll approach it. If you have some suggestion over top of this, you are welcome to give it. We can anytime modify our ways.
Yes, Prasoon.
I had a basic question. I don't know about the background of others, but I actually got the link for this particular course through my lab member shared on my like WhatsApp group, and that's how I got to know of this. So technically, I got into the field of cognitive neuroscience for like almost a year.
Oh, nice.
But then I decided to switch back to biology, which is my parent domain. And so when I was actually like interviewing for the course of my second master's at Washington State University, where I was doing the cognitive neuroscience work, so my PI asked me if I know MATLAB, and I was like, I am not very comfortable with MATLAB.
Okay.
And she was like, okay, I don't know Python, so you'll have to figure out the stuff. I collected the data, but I did not do the processing, and that's the reason I want to learn the processing part as well. I came back to India on a leave and then stayed back. So my thing is, why Python over MATLAB, and why people are still using MATLAB? Or like, how do we?
Okay.
Is one better than the other, or why?
Not really. Both of them has same capabilities, but the problem is MATLAB is only accessible through a license, but Python is an open source, and there are many libraries which very customized libraries. So that way Python is advantage, and it has like notebook kind of thing. But nowadays MATLAB also introduced live scripts. So the only restriction with MATLAB is it's not open source. Otherwise both are same, and in fact for signal processing techniques MATLAB is even more comfortable to many of the people. And yeah, so you have.
Do you use MATLAB for?
Yeah, I use MATLAB for all my analysis. Just for the sake of course, because it's an open source, we are moving to Python. But nowadays, like you can go hand in hand, and you can use various code or code pilots or ChatGPT. You give a Python code, it converts to MATLAB. If you give a MATLAB code, it converts to Python. Although you need to do some debugging, but end of the day, nowadays it is easy to switch between these high level languages. So yeah.
I did a 5 days course on MATLAB and I was like just done with it because I just didn't get it. It
Oh.
was too weird for me. So if someone just wants to stick to Python, is that fine? Will that hurt? I mean, if the ideal case situation, what would your recommendation go? Would it be MATLAB or Python?
Mm,
Anything works. Like in our lab also,
yeah.
there have been even like master students who are using MATLAB instead of Python, and they are developing it solo. Like since I have developed my notebooks in Python, my supervisor had already developed it in MATLAB, but I decided against it and I was using Python.
Yeah.
I was
Yeah,
not
my
familiar.
question is like, if like in the ideal condition, because primarily I am not comfortable with MATLAB at all.
Yeah, that's fine. Like you
So
can continue
will
using
that hurt
Python.
in
I
any
don't.
way, anywhere?
Oh, MATLAB anywhere? Yeah, like lot of lot of universities have access to this MATLAB, and but Python is also equally user friendly, and nowadays many of the people are shifting. So even like a lot of my lab mates, prior to this hype of Python and all this, they were using mostly MATLAB. But when the uh, like Python is open source and it has many libraries now, so they started shifting to Python, and also it's easy to convert between Python. You don't have to learn the language per se, but you can actually it's always syntax thing mostly, but the logic remains the same, so you don't have to worry about it. And for this course we'll be trying to keep it mostly Python, and nowadays there are add-ons on the Python add-on on the MATLAB, or MATLAB data conversion on Python. So that way, like you might use for pre-processing and various signal processing techniques, you can use MATLAB, and then for interpreting statistics, machine learning, you can use the same data set to the import to the Python and then use Python codes. So it is always interchangeable, but it's don't need to worry if you're uncomfortable with MATLAB. So we will mostly be using Python itself, and yeah.
Thank you so
Yeah.
much. All-
So any more questions in this tentative plan or timings of the course, etc.? Okay. So
Also, just one request. So I'm typically not from a mathematics background. I'm a core biologist.
got it.
So it would be great if you can just like slow down where there is like
Yeah,
complicated mathematics in terms
definitely.
of
Yeah,
Fourier
definitely.
transformation and all that
Yeah,
stuff.
the main goal of this course is to make everyone comfortable with, like, they should not be analyzing data and be thinking behind their head that it's all black box and I'm not able to understand just using the commands. So we will definitely make it simple and user friendly to help you guys understand the maths behind it, and we will not go too much into equations also, because the course has a population from diverse backgrounds. There are bioengineers, biostatisticians, electrical engineers, and even some MBBS graduates. So so we will keep it simple so that it is friendly to everyone. So you don't have to worry about not having math background after 10th, or not having coding background much. So, and also code books are we'll do hands on, so we will be doing together the code stuff, and since it is in notebook format, you have chunks of codes, so you don't have to make sense of all the like 300 lines of code, but instead we will be doing chunk wise based on the weeks in parallel. So that way we'll make it user friendly, so no need to worry about it.
Yeah, thanks for the clarification.
Yeah. Okay, so now we have done with the course logistics. If you have any queries, you can email to us or write on the Slack on the group, or anything works. Uh, just on the closing remarks of the first session, we just wanted to show what potential can understanding or interpreting this neural data lead to. So we will just show like few nice snippets from real-time examples using this neural data. So the first one goes here is about a high performance neural processes for speech decoding. So what if in a person's life, if a person loses his speech because of some accident or some muscular dysfunction, so how we can use electrocorticograph and microelectrodes array to decode the speech. And this is a multimodal data where they use muscular information, ECOG, multi-electrode array recording. So I'll just play it. Let me know if the audio is the audio audible?
Yes, yes.
Okay.
I'm Eddie Chang. I'm a neurosurgeon at UC San Francisco. My lab works on understanding how the brain processes language. One of our projects is to develop a device that can help people who have lost ability to speak to once again communicate. We've just published a new study in the journal Nature that builds upon our previous work, which showed that it's possible to record neural activity from a paralyzed person who is attempting to speak and translate that activity into words and sentences as text on a screen. In this new study, our translation of attempted speech into text reached about 78 words per minute. We also show that it's possible to translate the neural signals not only into text on a screen, but also directly into audible synthetic speech with accurate facial movement on an avatar.
I think you are wonderful.
This device reads the blueprint of instructions the brain is using to give to the muscles in the vocal tract. Our goal in incorporating audible speech with a live action avatar is to allow for the full embodiment of human speech communication, which is so much more than just words.
Believe me, it is better.
For us, this is an exciting new milestone that moves our device beyond proof of concept, and we think it will soon become a viable option for people who are paralyzed.
So here you see a like speech decoding from a yes. Jayan sir, you can please answer? Sorry, yeah, you can please
Unmute.
Yeah, unmute. Your
Hi, hi Deepak. It's good to see you guys.
Yeah,
You're doing fantastic job. I have a question regarding this kind of work. How do you authenticate the data? I'm sorry, I'm traveling, so there might be some disturbance. When you say that you are able to interpret the speech by studying the brain signals,
yeah.
how do you make sure that that is what the person who is not able to speak is trying to say?
Oh,
What is the control?
Acha, I
in this
see.
case.
So this person from who is a participant on this study was able to speak when she was in childhood. She's like her functions are fine except for her speech. So she can still make the speech movements where her tongue or the vocal cords move, but she can't make a speech which is audible to the people around. So what they did is, this is a multimodal data being sent to the deep learning model, where it is trained on her speech which was recorded when she was a child or before she lost her speech. So they have the speech data, they have they have muscular data because her muscles are still functioning but she's not able to produce the sound. And then they validate it based on her, like she can write, so based on her how she communicates generally to people, she can communicate to the people, and that they can validate it. And that is how they validate it based on the subject's questionnaire. But there could be better when they can decode from a human. But for a normal human, the cost here is we can't make an implant unless there is a consent given to us. So now, like, Neuralink is going to try on normal humans, where whoever can volunteer, they can volunteer and try to augment their cognition or speech. So yeah.
So it seems like there has to be some kind of a muscular movement, as you said. So take into account the muscular movement and perhaps what she, perhaps they knew what kind of answers they'll give, right? Because she wrote them.
Correct.
Something like that would have happened, because the point is maybe she was one of the less severe cases of, people who are lacking,
I agree, yeah.
or they've lost their, voice or something, right? But then what about many people who have a severe form where the muscle movement is not only brain functions?
True.
How do we talk about, how do we address the severity?
In these applications, I also think that not only severity, but like even if you get a similar patient, the model may not work on the next patient. Like you have to train it from scratch, or but so once you show that this kinds of model can work on one patient, then it gives hope to people that you can try it out at least for different patients, and this may one day, like even if you need to retrain the model, but these kinds of model can decode the speech from brain activity or muscle activity. So that is something fascinating about these, that it's not implanted in the muscles but in the cortex.
Close to the supplementary.
Exactly.
So that makes it more interesting, that you're not recording actually the tongue movement, but the brain activity, and you can decode speech and also movements from there.
Towards the later, they have used the signals of muscle movement or muscle activity of the tongue because they wanted to use it in an avatar which is more natural. So that is other case, but the prior study to this, they have a ground truth of what. So there is a prompt on top, and then they have to try to speak it, although they're not able to speak, and then the decoder decodes it, and then they find how accurate is the decoding. So that was their prior study. After validating it, they moved to this even advanced study where they actually convert into live speech using an avatar. So, and also there are different validation techniques in this data science or ML, where you have tests, and validation.
Yes, thank you.
So that's it. Any other doubts here or any comments? Okay. So here you see another thing from Meta Reality Labs, basically Facebook prior. So they have used these neural signatures are not only the brain signals, but also we can use has a rehabilitation mechanisms where people who have lost motor control, but they have still the signals from the brain to muscles are going. They can use an EMG wristband, and they can convert into different force levels, or handling a bionic arm or robotic arm on their side to assist them. So
with AR devices, we're asking the question, how do we build a computing platform that is truly human-centric?
Facebook Reality Labs is a pillar of Facebook that's dedicated to bringing AR and VR to people, to us, to consumers of the world.
Every single new computing era requires new input devices, the set of interactions that really make this possible.
With AR glasses, I think that the key here is to communicate with our computers in a way that is intuitive at an entirely new level. The wrist is a great starting point for us technologically because it opens up new and dynamic forms of control. This is where some of our core technologies like EMG come into play.
Neural interfaces, when they work right, and we still have a lot of work to go here, feel like magic.
So if you send a control to your muscle saying, I want to move my finger, it starts in your brain, it goes down your spine through motor neurons, and this is an electrical signal. So you should be able to grab that electrical signal on the muscle and say, oh, okay, the user wants to move the finger.
What is it like to feel like pushing a button without actually pushing it? And that could be as simple as, hey, I just want to move this cursor up or move it left. Well, normally I would do that by actually moving. But here, you're able to move that cursor left. And it's because you and a machine agreed which neurons mean left and which neurons mean right. You're in this constant conversation with the machine.
This new form of control, it requires us to build an interface that adapts to you and your environment.
Everything starts with a click.
The intelligent click is the ability to do these highly contextual actions in a very low friction manner.
It's kind of the purest form of a superpower.
The wrist can also be a spot where the technology is communicating back to the user.
The haptics, the sensation of touch around us, this is part of how we learn and use motor control. It's critical to AR and XR.
We wondered, as you pull back the bowstring on a bow, if we tied that not to the tension growing on people's fingers, but rather squeezing on the wrist, would it add to that experience like you're pulling back the bowstring? The answer is yes.
That future, it really is the computer that is seamlessly integrated into your day-to-day life. The next computing platform is the mixed reality platform, the one that really totally blends your virtual environment and your real environment in a seamless way.
We're in this moment where we can move from personal computing to personalized computing.
What if you and a computer agreed to design a keyboard together, and you type faster on it than anybody else in the world could type on your keyboard?
I think what this enables is the ability to not have to focus on a computer, on a phone, and be able to still interact with other people. It's going to open up a new generation of communication.
Okay, so this is a very interesting thing they're doing, and they also not just enabling you to augment your motor movements by not actually moving, but also giving you the feedback, haptic feedback, so that you have exact sense when you actually move, or when you actually, how much force you apply on an object. You get a feedback, right? You always have a sense of how much force you're applying. And there should also be a control where you just can't use a bionic arm and just hold an object and it just crushes it. So if it's a glass, you need to put certain force. If it is a sponge, you need to put certain force. So that way, these technologies are very interesting, and this is only being enabled by the new recording techniques, new data processing, statistics, and decoding techniques that are available, and the research going on around them. So using this course, you'll also learn some, not exactly implementing the whole end-to-end system, but basics of how to deal with the neural signals. And in your future or in your projects, if you want to implement some part of this big projects, you can always make use of this basic processing. And the time you consume if you don't take this course, or if you don't know the processing pipelines, then that time would have reduced, and you'll also be more confident and comfortable to apply. Yeah.
So these kinds of applications require them to segregate. So lots of muscles go from your hand and lots of nerves go from your hand. So how do you decide with pinpoint which muscle is exactly being activated at that particular time? So the analysis is basically an exaggerated version of what we'll learn in this course, that especially in the electrophysiology part, that how do you find out which particular unit is spiking. So if you know that, okay, these kind of things I have learned, then this is just a much more advanced version of that only, and I can do that. So that kind of feeling we should get after this course, I guess.
Yeah, hopefully. Where is this?
I have to click here, I guess. Now do.
With it.
Yeah. So so we have seen in human models, but we also need mice models, because in human models we can't do invasive studies. So we need mostly non-invasive recordings like EEG, MRI, which I will be dealing. But the advantage with animal models is, or from primates or rodents, is you can analyze them at the neural levels and at more depth and more region levels, which would help us to understand the mechanisms behind same parallel phenomenons in humans or other organisms. And yes, So we'll see a thing called grid cells where okay, so Prasun says they use organoids where okay, we can discuss this later, but organoids are you create from cells you make a organoid type of brain where it is a population of neurons, and then you can directly analyze on that rather than using animal models. So that is also an interesting part. Maybe Prasun can mention after this thing. So here we'll see how it's called internal GPS. How do we make sense of space? So there are certain population of cells which always fire when you reach certain positions in a space. So your brain already has a map of your space, and then you can see how it goes. So the red dot represents how well a population of neurons firing, and you can build a grid out of it. So this is a mice which is moving around. You see the movement being tracked, and the red dots wherever they come is when certain set of neurons that you're recording fire. So a population, or let's say you can take a single neuron also, it fires all the time whenever it reaches certain positions in the grid. It fires more than baseline. So when it moves, you make a map of it, and then you see how the grid forms. So this cell responds to these particular positions, and like that you have either multiple neurons or multiple populations across which okay, just a second, where you all the populations integrate together and give a sense of your position, or there are also head direction cells in a similar fashion arranged. So these are the cells in your hippocampus, which is mostly useful for your memory and long-term memory, and also used for spatial navigation. So, and it has been found that this thing is conserved across primates, mammals, and various species also. So that way you can translate from animal models to human models as well. So Prasun, oh yeah, Jayant, you can yeah.
Yeah, I just wanted to know a little bit more about this particular experiment that you have shown,
Yeah.
where you're trying to capture a specific region and you're just trying to see which of the neurons are more active compared to others.
Yes.
So, what kind of activity were they recording? And
so this is the electrophysiology.
do you think that the activity that you're doing, either reading or writing or perhaps watching a movie, can actually change what kind of neuronal hubs are more active?
So these are called grid cells, which are mainly present in entorhinal cortex, and which is like directly connected to the hippocampus CA1 region. So these are electrophysiological recordings where you record the spikes from a depth electrode, and as you can see, here these are the spiking activity, and
from 4 different units.
Yes, and they put a threshold on it. Whenever this from 4 units you get a spike, it will make you'll put a red dot here. So it is like a thresholding. Whenever the activity spike crosses a certain threshold, you put a dot. So it's like for that particular place in the space, that particular neuron fires. So either if you take calcium imaging, it works like the neurons give a fluorescence out of it when they fire. So that activity also is correlated with this activity. So most of these recordings have correlated responses, but we have to take with a bit of grain of salt, because there are certain spatial and temporal trade-offs, as we have seen before. So if we encounter for that, for example, if you're seeing a response in certain region, certain electrodes from EEG, and also your similar experiment, you're testing on an MRI scanner, under an MRI scanner. So the MRI scanner timescales are very slow compared to EEG timescales. So if we can do, there are methods called deconvolution methods in time, where you can have an assumption of a MRI response with respect to EEG, and you can compensate for the delay, and then you can make sense of it. So there are certain processing steps across domains if you want to see the data, but mostly these are like this analysis particularly is just firing or how active is a neuron. So that should be same across whether you use optical fiber photometry or electrophysiology. So, does that answer
I agree with you. My only point was that since I'm not a neuroscientist, yeah. So, but I'm trying
Sorry, we lost your.
is that what I see on the screen is a set of neurons which are firing more compared to the neighboring neurons, right?
No.
So you see some of the hubs over there, right?
No,
which
no,
are
no.
deeply red, and then you see some hubs which are, maybe.
So what you see, the red portion, the map
Yeah,
is
please continue.
the physical map. Basically, in the space here, the animals, the traces you are seeing is the animal moving.
It's only the movement traces here. It is in the
So-
space, but the dot only represents how whenever this four neurons.
No. He's saying that these are neurons. So
No.
So what it is that on the left, it is an open field experiment where the animal roams around in an open area, and whenever it while it roams around, you record from neurons. So from the recording, the scientist took identified these 4 particular units that fire specifically in these red zones. Whenever the animal is in these red zones, the neurons fires. And when you take out on an average, when the neurons, these particular 4 neurons fire, they fire in a grid pattern.
Yeah, this is a heat map showing the heavy firing regions versus the light, the cool color ones are less firing regions.
So these are called grid cells. That's why.
Yeah. So this particular 4 cells respond to these areas more than any other region in the space. So this is a map of space and not the cortex, not in the brain. Does that answer, Jain sir? Okay.
Right. So I did not understand the experiment. Now I do. Thank you.
Okay. Sure. Cool. Okay. Prasad has something to tell about organoids. Should we ask?
We'll finish it first.
Okay.
And,
1 second. Okay. I think we'll skip this. So here is another recent study where they are decoding semantic reconstruction of continuous language. So what happens here is the subjects. 1 second. Yeah. So the subjects are when they watch a movie, their fMRI responses are recorded with audio, and then they train a model which learns on their fMRI response and map to the imagined speech or semantic interpretations. And then you can see later on the test when they show a movie without any audio, and the MRI responses go into the trained model, and it will predict the semantic representation of that particular scene here. So this is another interesting study where MRI responses are decoded to the semantic representation of certain scenes here. So there is certain level of errors happening, but still this level of decoding is still good, as you can see, and you can go through this study and get to know more about how the error rates of this model are. So, but this is just the beginning of, with a non-invasive study, at that resolution, you're able to decode the semantic information from the brain. So this is another interesting study, and this is a model they used. So I'm not going into depth in interest of time, but you can always go through this study, or we can discuss offline if anyone has any inputs here. So just want to end on talking about positive feedback loop of neuroscience. So here is the neuroscience where we try to understand the brain and make sense of neural data. So with advancements in imaging technologies and high data rate communication technologies, data storage systems, neuromorphic systems, we are able to image, store and analyze this neuroscience data, and in turn studying this is also helping in a positive feedback loop, that it's always improvements in technology improves our understanding of the brain, and our understanding of the brain again helps us in making neuro-inspired chips called neuromorphic chips, which are a very interesting thing where they mimic the synaptic activity on the uh, electronics chips, which can have more efficiency and more sparsity so that they consume less energy. And we can also use in augmenting cognition such as brain machine interfaces or brain co-processors where a function which is not present or gone over time because of some disorder or some accident can be put back into the brain where for example a particular brain has a lesion because of which the signals are not being communicated across. So what we do is we put a co-processor where neural signals are recorded by that co-processor and then it re-simulates in the other region. So it bypasses that lesion and then writes the signal to the brain. So that way it can augment cognition. It's still in the beginning stages, but these are some of the advances or research where it is happening in terms of this domain. And also advances in signal processing, machine learning, deep learning helps us to interpret the neuroscience data better and make good discoveries or good diagnosis. And this again, understanding the brain helps us to make neuroscience inspired AI or biologically plausible learning, which is more efficient than which is known to be more efficient than general training of this very deep neural networks. And also how to develop energy efficient algorithms, because as we have discussed, brains are very energy efficient. Same computer vision task which takes a lot of energy, for us it is very granted. It's like it's in very less delay and it takes very less energy for us and it's very intuitive. So on this positive feedback loop, we want to end this session. If you have any comments or you want to discuss anything, please let us know. Yeah, sure, we'll paste the link. We'll send you a mail directly, so
you can take the screenshot.
Yeah,
you can take the screenshot and then join from here probably.
Yeah, but I think okay, we can share. This is for the course. Yeah, you just go through IEEE course online and from there you click on the neuroinformatics course. But in case you're not added, we have to wait for some time because we need to get a confirmation from the team which is helping us to organize this course.
Also there is one more Moodle link that will be floated.
Yeah, also one more link will be floated and you can join that also if you're not being added to this group. Yeah.
Okay.
Yes, sure. We'll upload and we can also share on Slack so that who don't have access to Moodle can access to that. Yes.
Do you want them to introduce or?
I think it's already 7:55.
But I think that will be useful.
Yeah.
For us. So if yes, you have you want to introduce yourself for your course mates and to us also.
Yeah.
So that we know your backgrounds. It will be you. And if you are planning to attend the course regularly, or do you have any specific requirement like interests that you are targeting, that I will attend these particular lectures, then you can tell us. So that we get an idea from.
Yeah, you can keep it short and introduce yourself.
Yeah, because we are already short on time, so very brief.
It would be good if everyone can do it, or if you're uncomfortable, people who are comfortable can do it. Yeah, one of them can start and then.
Yes, should I go?
Yeah, sure.
Yeah, so this is Vinod Kumar. I'm a research scholar at the Center for Neuroscience. Where my expectations from the courses are that I want to be a little more sort of better at coding in general. I not use Python, so that will be good. And so to get a little more skills on signal processing, some introduction to some sort of ML, DL techniques, if possible.